All right. All right, let's start record. Where's my record button? Okay, guess we are recording. All right, let's go. Let me do the share. Let's start here. Let's go here. Edit the PowerPoint so we can get it in screen view. And let me find out where we left off. Hold on. This is part 2 of chapter 14, the autonomic nervous system. Covered that. All right, this is where we're going to left off. Remember where we're at. Try to remember where we're at. We are, if we zoom out of this right here, we are in the, you have our central nervous system, and then we have our peripheral nervous system. So we are in the peripheral nervous system. Then we are going into the autonomic nervous system. So we have the ANS, it's right here. And of the ANS, we have the parasympathetic and the sympathetic divisions. No matter where we go, what we're talking about, we're gonna be in the ANS. And within the ANS, we will be somewhere in the sympathetic division or the parasympathetic division. Just wanna say where we are, right? Sometimes we're gonna be looking at a lot of people, we're gonna be seeing a lot of CNS, like brain and spinal cord, but that's to be expected because the wiring has to connect to, remember, we are peripheral to the CNS and has to communicate. So it has to be at some point rooted right here at some, it just has to be. Now, speaking of this, the sympathetic versus the parasympathetic, the sympathetic is more complicated. The wiring, the hard wiring of the parasympathetic division is much more complex than the parasympathetic division, okay? Now, if we can take a look here, from T1 to L2, we have a multitude of abilities to control. And again, this is autonomic. Therefore, this is involuntary. Involuntary of, right? We have we have visceral organs and cardiac tissue. So, so smooth muscles and cardiac tissues. So we have things like, okay, up in here, this region up here, we're looking at eye, mucosal. We then get into salivary glands, heart, lungs, liver, stomach, spleen. All these areas, if there are issues here, we will have issues with these things here. So that's just the nature of the situation. So as we're take a look at T1 to L2. Now, in terms of the pathways of collateral ganglia, they will most likely be from, that's why, or most fibers from T5 to L2 synapses. And that's the range in which that's going to take place in. Not gonna ask you on that. I'm not gonna ask you those type of questions on those type of is it gonna be from T5 to L2? Not gonna do that. Um, but I just wanted to give you a heads up here. Okay. I just stopped it because I saw blinking of the chat. So I just wanted to make sure maybe I wasn't recording, maybe something's happening. So I just saw the blinking and I just stopped it to see where we were at. Okay. Now, please keep in mind that this jumped. So I need to move this slide here. So the passing through sympathetic trunk to the synapses of the collateral ganglion anterior to the vertebral column. You may be wondering, where does this go? I was playing with the slides. I must have moved it, but this goes over here. It goes back over here. And I'll move it for you. Where is it? Here. So here's 1, here's 2, and then we have this in between, and then we go 3. So I'll probably be moving it. So we have we have to communicate, right? So when we see this here, this is again our gray matter or white matter. It has to link up and it has to communicate. Remember, afferent, efferent, two-way highways, right? We have to go from some site. In this case, let's say cardiac, have to go from cardiac to CNS, from CNS to cardiac, because we can't be constantly thinking about pumping our hearts. That's just too much, way, way, way too much to go on. So this has to be in the background, all by itself, running the analysis to see if your blood pressure is doing okay, your blood sugars are right, homeostasis of all your other organs. All this is happening. If you had some pizza today, it's processing that. You don't have to think about it. It's already doing it by itself. It's pretty cool, and it can be a little on the complicated side, but it must be communicating with each other from receptor sites to the CNS. But we are not in the CNS. So again, don't get that confused because we see brain and spine. Okay. Let's see. No, hold on. Now let's talk about pathways with synapses in the adrenal medulla. And I want to pause here because we're going to be discussing things like some important topics like norepinephrine and epinephrine and some of the things that's going to be happening. Well, we saw back here, and we know that there is the adrenal gland right there, right? Okay, so it's between the T1 and L2. Got it. Again, I'm not going to be asking you where that location is, but I will ask you things of this nature. So some preganglionic fibers, remember, we have the pre and we have post-ganglionic fibers. In this case, the preganglionic fibers pass directly to the adrenal medulla without a synapsing, right? Without synapsing. So what do they mean? Well, you can see here that they're not, it doesn't have the synapsing compared to other parts. So we come back here and say, so what is one of the effects of CNS to the adrenal medulla? Or in other words, what are some of the effects of the adrenal gland? So when stimulated, not inhibited, right? The opposite, when stimulated, medullary cells are gonna secrete norepinephrine and epinephrine into the bloodstream, right? So sometimes some people call it the noradrenaline or norepinephrine. Remember, if we have a stimulation We must have an inhibition. You can't keep secreting this stuff, right? That would not be good for homeostasis. This is done for specific reasons. It must be balanced. It doesn't say it here, but I'm telling you, the same wiring has to send signals back to the adrenal medulla saying, hey, let's stop that action now. We're good. Okay. So just making sure we understand that. So sympathetic ganglia and adrenal medulla arise from the same tissues, right? So one, it could be considered that the adrenal medulla, this is just like a fun fact right here, this last bullet, that the adrenal medulla can be considered misplaced sympathetic ganglia. Not going to ask you that stuff. What I want you to know is these 2 bullet points up there. And here we take a closer look, right? We're taking a closer look at the adrenal medulla. So we have here this are going to be our spinal cord cross section. Therefore, now we go peripheral of the spinal cord, and specifically, we're just looking at a T8 through an L1. Again, not going to ask you those type of questions, but we have this running through. We have our sympathetic trunk, and we have our nerves. Okay, well, here's the kidney, and above the kidney is zoomed in, and we have the adrenal medulla. Within the adrenal medulla, let me zoom in for you a little bit. Within that, we have these adrenal medulla cells. Well, look at that. It's kind of coming in here, and they're not really, we won't get into the biochem, but we have a receptor right here in the end. And based on the signal that is received from your CNS, specifically spinal cord, right? And also brain running down, also spinal cord communicating to the signal. And we have now what? We have these cells releasing epinephrine and norepinephrine via blood vessels. So super cool stuff that's taking place all the time without us knowing, because we can't manage all these tasks at the same time. Just very difficult for us. What about these visceral reflexes? Let's take a look into these. Well, in order to have a visceral reflex, you must have a visceral sensory neuron. So you have to sense something. If you're going to be reflexing or reacting to something, you must sense. That is the beginning pathway, right? Remember those, the ABC, remember the 1, 2, 3, 4, 5s. So we have to sense. All right. So therefore we have to have sensory neurons, and they're going to send information about chemical changes. It could be a stretch. It could be, when you mean by stretch, it means an actual physical stretch. It could be that. It could be different chemical. I don't like that word, chemical changes, but it could be the biochemistry that is happening in your body, the biochemistry, biomarkers, right? Not really chemical. Again, we keep seeing this over and over and over throughout this entire course. It's the chemical. No, it's the biochemical. Temperature, absolutely. So these things are going to be important in order for us to react to them or reflex to them, right? So receptors, now we're getting so sensory, then on receptors. So receptors for the visceral senses are free nerve endings, widely scattered throughout the body. You must have these free nerve endings to be able to get information. Let's say I touch something, it's hot. I need to know how to react to that. I need to know that I can move back and get away from that environment. Or if I'm standing in the sun, it's a little bit too hot for me. It's just kind of like burning my skin. Okay, I can move this way. Those are possibilities. But all that there that I just mentioned, those examples, those would be things that we're going to be sensing. But what about we're sensing and consciously mediating? But we're in the autonomic system. So I mentioned, right, that this is doing stuff in the background. So what's going to happen? Those are things we're consciously doing. What would be an example of temperature that we're not consciously involved in terms of temperature going up or temperature going down? What would one be?
Fever.
Yeah, fever. Great. That's a perfect one, right? So you have to communicate. Then we get into the immune system and all this. And then we get our thermostat of our in our brain, and it helps us regulate temperature. So we have to have sensory and we have to have receptors in order for this to take place. In the case cell bodies, remember what those cell bodies are, right? Your cell bodies, we're talking about neurons. Don't forget what a neuron, typical neuron looks like. They can change. They can be some of them in the parasympathetic versus the sympathetic. Some of them have more myelination, some can have less myelination, so forth, right? But still, remember what a cell body is. Cell bodies are located in the dorsal root ganglia and in sensory ganglia of cranial nerves, right? So axon, remember what the axon is? That yellow line, right? The yellow line that stems from the cell body. Axons travel in same nerves as autonomic motor fibers as they monitor conditions or organs serviced by the autonomic nervous system, nerves. So those axons still come into play. What's really interesting is now we are in chapter 14 and chapter 11 is still heavily, we cannot shape away nervous systems and nervous tissue. We just can't get rid of it. It's here to stay. That's why we took a lot of time on it. The visceral sensory neurons can converge on the same second order sensory neurons that can receive input from somatic sensory neurons or play a role in referred pain. Very important stuff. Very important stuff. One of the case studies I wanted to do, but I was giving you quite a bit, was the individuals who cannot sense, have no visceral sensory neurons for referred pain. Very rare. I didn't want to throw it out there due to its rare situation, but it does happen where some people can put their hand on a hot surface and not know it until they smell their tissue burning. Very rare, but it does occur. So in terms of visceral reflex arcs, right? So we have the same components as a somatic reflex arc, the same main components. You have to have a receptor. You have to have a sensory neuron. This is what I was trying to tell you about the ABCs or the 1, 2, 3, 4, 5. So we have the receptor 1, you have the sensory neuron 2, you have an integration center 3, you have your motor neuron 4, and you're going to have your effector, your 5. So that's your 1, 2, 3, 4, 5. That's what I was trying to help you with. And that is that if you're wondering what is he in the heck is he talking about, that's your chapter 13 lab exam review. I still need to post that. I need a video. I already have a video for you. I need to post that up. But if you look on your PowerPoint slides, you'll see and know exactly what I'm talking about. So the 13 comes into the 14. So it's very important. So we have here, let's take a look at bullet number 1 here. Visceral reflex arc has 2 consecutive neurons in the motor pathway. Afferent fibers. Remember what afferent is. Don't get it confused with E-F-F-E-R-E-N-T, efferent. We're talking about afferent, right? So that means going to CNS. So fibers are visceral sensory neurons. And that makes perfectly sense. Effectors are the smooth muscles, like I talked to you about. We said smooth muscle and cardiac muscle and glands rather than the skeletal, because you did not hear me say that in terms of, I did not say skeletal. I specifically was saying smooth and cardiac and glands. What would be an example? Reflexes that empty the rectum or a bladder. So these are one example of a visceral reflex. So you have a 3 neuron reflex arcs exist in the walls of the gastrointestinal tract. So as a result, they have to have sensory neurons, interneurons, and motor neurons. They're going to be involved within this. So diet is very important for things of that nature, like gastrointestinal tract and health and all these fiber and all that stuff, colon health. All these things are great, great things that we must try to do our best to maintain. But there are some things that are beyond our control. And I think we're kind of getting a hint of that in what we're saying, meaning a lot of the stuff we're talking about is beyond our control. It's we're already wired, and if we have an accident and they affect the wiring, then certain things can happen. It can affect bile movement. There are many, there are different outcomes that can happen. So genetics comes into play, and then we have potential diseases that we classify as diseases. If we have any type of neuron degradation, the wiring degradation of it, we have a problem, Houston. We have a problem. So we can see how important our CNS is to our PNS, and how important our PNS is to the rest of our functioning bodies. They have to go together. One can't be better versus the other. They must be checks and balances. And that, as we study more, and as you get into, say, micro, and you get into all these other classes and you start learning more and more, I hope you find a moment where you have a serendipity moment and you realize, wow, what an amazing organism we are. We are very complex. There are a lot of things that it's doing in the background that we don't even have control of. And this is one of the reasons I say that one of the biggest tragedies of life is having a perfectly healthy human do nothing with that life. The reason I say that is because I understand the background. I can see the miracle behind it, a healthy human being. And I use the word miracle purposely because it's very complicated for me to just kind of just put it mathematically. I struggle with the math. So I then say, very interesting that so much wiring has to take place and it has to be just right. So it's a very interesting thing, and hope I'm reaching some of you. There's the 1, 2, 3, 4, 5 that I was talking about. So please make sure you guys get this part, and in terms of visceral reflexes. So, and then we have, again, a stimulus, going through the 5 steps, and then we have our response. All right, so just make sure you guys get that down. Let's dive into a little bit of neurotransmitters. Remember, there's something that we need to talk about before we go into neurotransmitters. The way we are communicating, no matter how you want to talk about it, we are communicating in terms, not vocally, but in terms of what's happening in our bodies. Communication from CNS to PNS. It's only done 2 ways. It's only done by biochemical neurotransmitters or via some action potential, right? It has to be one or the other. And just going back, we discussed that those 2 major methods of communication. The neurotransmitter is, of course, slower. We know that. The biochemical route is slower than the electrical route. Keep that in mind. We're going to talk a little bit about neurotransmitters. And again, we will stop the lecture at 7:30. And then we'll come back. Okay. All right, neurotransmitters. So major neurotransmitters of the autonomic nervous system are acetylcholine, norepinephrine. They're abbreviated ACH and E. Just try to remember the abbreviations. Our presentations here made by Pearson, they love to use the abbreviations often. So make sure you know ACH is the acetylcholine and NE norepinephrine. Very important. So I'm focusing here. I'm right here. Acetylcholine is released by cholinergic fibers. So all ANS preganglionic axons, all parasympathetic postganglionic axons. How about the NE? NE is released from not cholinergic, and look, here's the key, guys. Acetylcholine, cholinergic fibers. So this one and this one go together, right? Now, in terms of the norepinephrine, that one's going to be the adrenergic fibers. Got tongue twisted there. In this case, think of this as adrenaline, but in this case, we have norepinephrine. So almost all sympathetic postganglionic axons, except those in the sweat glands, release acetylcholine. So it's widely, if you have to choose one of the two in an exam. Let's say you read a long, one page. I throw a one-page question at you, and you're like, you are struggling for time. And you're like, I can't read this one-page question. And then it has a multiple choice between ACH and norepinephrine. You're probably best off just randomly picking the acetylcholine because of this thing right here. Just keep that in mind. So effects of neurotransmitters depend on where it binds. Where is it binding to? Which receptors, right? That's what matters. So the effect will depend on its receptors and where it is going to bind. Let me try and move the slide. All right, let's just take a quick look at this. I did this in the other presentation, but I'll talk about it really briefly here because it's really important. Autonomic nervous system, right? Breaking up into the parasympathetic, sympathetic, and here we have the somatic nervous systems, right? Because when they're in parallel, they're side by side, but this is the one that breaks out into the para and the sympathetic. Briefly, I already discussed this before, but we're in this presentation now, and I'll just do it quickly. So you want to look at this. This is a wonderful presentation. I like it. I love it. So let's take a look at the somatic nervous system, and they have one neurotransmitter, acetylcholine. Got it. Acetylcholine is going to be a stimulatory on what? Skeletal muscle. So somatic nervous system, skeletal muscle. That is how come we and our eye did not discuss skeletal muscle, because we are in the autonomic nervous systems, and specifically we were talking about the sympathetic nervous system, sympathetic division. So that is why I did not mention this, because it does not take place here. Okay, let's take a look at the neurotransmitters. Hey, guess what? We just saw that. Acetylcholine, norepinephrine taking place. And because they are two differences, they are going to be antagonist of each other, or they're going to be stimulatory and they're going to inhibit, meaning they're going to have a nice control of one another. So you have the positive here, look, acetylcholine is positive, but you add the norepinephrine and you can have the negative. Again, the effects depend on where these two neurotransmitters are binding to, i.e. the receptors, right? Okay, let's briefly look at the axons. All right, axons of the somatic, they're gonna be thicker, heavier myelinated. Axons of the parasympathetic and the sympathetic, they're gonna be lightly myelinated. Just wanted to go through this with you here. We already did in the part one, but it's just a recap. Okay, mentioned receptors, and in terms of having effects. So we have two types of cholinergic receptors that bind to acetylcholine: nicotinic receptors and the muscarinic receptors. And You probably look at this and some of you may be like, wait a minute, these things kind of sound a little familiar. Well, they're named after the drugs that bind to them and they mimic acetylcholine effects. So nicotine and muscarine, like the mushroom poison. Let's say nicotinic receptors, right? Number one there. Where are they? Where are they found? Okay. On all post ganglionic neurons of both sympathetic and parasympathetic. That is how come I'm trying to tell you that if you have to pick between ACH and norepinephrine, and it's a random, you don't know the answer to, I would just pick that one. Also, where else can they be found? Well, on the adrenal medulla. That is where the receptors can be found. Very important. Now, remember the good old skeletal muscle chapter that we talked about? Sarcolemma of the skeletal muscles cells at the neuromuscular junction. That is so muscle chapter right there from AMP1. Effect of acetylcholine on nicotinic receptors is always stimulatory, positive sign. The effect on acetylcholine at nicotinic receptors is always going to be some sort of stimulatory effect. And what could this be? It could be opening ion channels. Yes, the good old ion channels. Like we go back into chapter 2, chapter 3, and we really talked about those gap finders of chapter 2 and chapter 3, ion channels. Very important, because remember, it is within those ion channels that things are going to be leaving and entering cells of the plasma membrane. What about this muscarinic receptors? Where's this at? Well, they're going to be found on all effector cells stimulated by the postganglionic cholinergic fibers, right? Postganglionic cholinergic fibers. What is the effect? So effect of acetylcholine at muscarinic receptors. In this particular case, it can be both an inhibitory or excitatory. Remember, think of homeostasis, meaning when you see this, meaning stop, go, go. Stop, inhibitory, excitatory, go. And it depends on the receptor type of the target organ. Which receptor is it binding to? For example, binding of acetylcholine to cardiac muscle slows the heart rate. Okay? Binding of acetylcholine to cardiac muscle will slow the heart rate. Whereas binding to intestinal smooth muscle cells is going to increase mobility. Don't worry about these. I'm not going to get into the receptors. I'd love to, but we're going to pass. We have enough information to keep us busy. We won't get into all this, but just know what's really important. I want you to make sure you understand this part is that we have different receptors. We have to. You have to. You cannot have neurotransmitters and then they all bind to the same thing. That would just be really weird and the same thing would always happen. We always have to have something that's going to inhibit and something that's going to excite. We have to have balance. So just think of it in those terms. There must be a balance. Neurotransmitters are the biochemicals of communication, so they're going to go around to a lot of different places. And depending on where they bind, certain things are going to happen. So the effect of a neurotransmitter depends upon where they land and bind. Okay? So that's the principle here. That is the principle. 10 more minutes and we'll take a nice break. So don't worry about alpha and beta. Just remember what I said. What I said is more important than the alpha 1, alpha 2, beta 1, 2, and 3. Don't worry about that. All right. Now this one here. Let's see. Now, let's go. We talked about acetylcholine binding to cardiac muscle receptors, slowing it down. Well, there has to be there has to be the opposite. If we're going to slow down the heart with acetylcholine receptors binding to cardiac tissue, cardiac muscle, there must be something that's going to do the opposite and increase its activity of cardiac muscle. What will that be? Let's go check it out. Norepinephrine binding to cardiac muscle receptors. Don't worry about the beta, right? Okay, so yeah, norepinephrine binding cardiac muscle causes an increased rate. That does happen. Look at this one. Whereas epinephrine causes bronchial relaxation when bound to certain receptors, right? So make sure we get this top one here, right? Which one causes the heart to go up? Which one causes it to go down? This is a great table. Table 14.3 shows you the receptor type, and it shows you the effect of binding. So it's a nice little table that puts it all in a nice order there. So if we take a look at some drug classes, right? That's not what we're talking. 14.3 talks about cholinergic and androgenic receptors in terms of the neurotransmitter acetylcholine and norepinephrine. However, table 14.4 is not this. We're now looking at a particular classes of drugs. Okay, so for example, we talked about, well, if you have like myasthenia gravis, right? Well, what happens is acetylcholine esterase inhibitors. So you can have an inhibitor that will inhibit the acetylcholine esterase enzyme. And as a result, you can have an increases availability of ACH. So there are some very, very cool different things that happen here. Anybody who has, we talked about lungs, right? We talked about in terms of well, we haven't really gotten into it, but like bronchioli or alveoli, things of that nature. So anybody has any type of asthma? Great, wonderful. No asthma, lung issues. Wonderful. Well, then check this out. There's a drug called albuterol. There are some other forms and names of it. This is more the what happened? Just trying to give you a 101 on some pharmacology. All right. So I'm going to talk briefly about, let's say, albuterol. Well, what's going to be, it's a clinical application in like asthma. So it dilates bronchioles by binding to a beta-2 receptors. Well, what is that? What's happening there? You have, look at your receptors, your androgenic receptors. And so it's going to enhance sympathetic activity by binding to androgenic receptors or increasing the norepinephrine release. These happen for both phenylephrine. Look at that. That's an active ingredient in nasal decongestants. So we have these things, okay? These things that take place. And it all comes down to these receptors. So let's make sure of the autonomic nervous system. Now, I'm going to be asking you some questions here. So table 14.4, I'm going to be asking you some questions. So for example, look at this one. Nicotinic agents, receptor bound, nicotinic acetylcholine receptors of all postganglionic neurons and in the CNS. What is the effects? Typically stimulates sympathetic effects blood pressure rises. Example, AKA nicotine, right? So these are some very important things. Hypertension drugs, also known as beta blockers. So just take some time to look into these, and I will be asking you in terms of maybe the drug class, and then its receptor, its clinical application, and its examples. So this is for your chapter 14 ANS. Again, as I was trying to tell you earlier, we're looking at parasympathetic and sympathetic. And we're taking a look at this, and yeah, it says most visceral organs have dual innervation, of course. So they're going to have these interactions. Action potentials are going to continue to fire down axons of both divisions, parasympathetic and sympathetic. And they're producing a dynamic antagonistic interaction. So antagonistic to one another, which does make sense and helps. Both ANS divisions are partially active. So that means, remember, the ANS breaks up into the parasympathetic, sympathetic. Well, both of them are partially active, resulting in a basal sympathetic and parasympathetic tone. What the heck do you mean? Well, they're both going to go like this. One or the other is not more intense. It doesn't typically work like that. They take turns. ANS, autonomic. There has to be balance to the system. Therefore, we have a parasympathetic. We have a sympathetic. And then we have certain neurotransmitters that are inhibitory, stimulatory. So as a result, if you were to somehow graph activity of parasympathetic and sympathetic, you would see both lines moving kind of like in a wave, a tone, like a frequency moving up and down in terms of activity, because you can't always stay intense in one division or another, or like turn off one, ramp up one. No, they kind of go up and down, up and down, up and down, and a partial activity part. So that's what's happening in the background of you. And let's see, where did we leave off? Let me minimize you. Oh, Zoom. Slide 61. Slide 61. All right. Well, everybody, let's see. I'm going to pause it for here. This will be the end of the part 2. This will be end. Actually, not pause it. I have to end it. This will be end of part 2. I have to wait for this to get loaded up. Then I'll head out to my office and we will wrap up the last portion of it. Okay. So take a nice 30-minute break. Come back 8:05-ish. Right. Come around at that point. And we'll wrap up the rest. We're at 61. I think it had like 86 slides or 84 slides. I don't remember. I don't look at, try not to know how many slides there are in there. And we'll come back. So have a snack, have some water, stretch or something. I'll take care of this and I'll see you in approximately 30 minutes. Okay. All right. We'll see each other soon. Bye-bye.